Thank you so much for being here. This is honestly the biggest Graduate Student Association turnout we've ever had, so thank you very, very much. So on behalf of myself, Rogan, the president of the GSA, and the rest of the Graduate Student Association and the Graduate College of Life Sciences, thank you for being here. All these students today that will be presenting have worked super hard for a really long time on something that is very, very hard for scientists to do, which is to communicate to the public. So that is what we're going to be doing today, and we're really excited to share this with all of you. So as I said, this event is presented by the Jefferson College of Life Sciences Graduate Student Association. Many of you have done Graduate Student Association events before. We're really happy for you to be here, not for a social event this time, but for something very, academic related. This is really important for careers and stuff. So thank you for coming. We would like to thank our sponsors that have made this event possible. This could not have been put on without the support from the Jefferson College of Life Sciences, which contributed the funds for the awards that will be presented to the students today, as well as the Jefferson Humanities and Health and the Office of Student Life and Engagement. So a little bit of background. Who here has heard, raise your hand if you've heard of the 3-minute thesis competition before? Okay. So for those of you who haven't, this competition is actually an international competition. It was started in 2008 in the University of Queensland in Australia when a professor was taking a 3-minute shower because they had a drought and realized when the egg timer went off that it would be an amazing opportunity for students to just present their research in 3 minutes. Since then, that event has grown, and now over 600 universities worldwide and over 65 different countries are putting on these 3-minute thesis competitions. So we're really, really excited to host one at Jefferson. All right. Oops. So what did our 3MT look like? So this started back in February 14th was our information session, and since then we had 2 3MT prep workshops, which were attended by 30 students. We had the preliminary 3MT preliminary heat, and we'd like to point out special thank you to our preliminary heat judges. We also presented our work on this entire project for the entire semester of our Science Communication and Outreach Initiative to the at the experimental biology meeting in Orlando, Florida, which happened a couple days ago. And it was widely attended by many, many people from around the country and world. Lots of people were very excited about what we're doing here. So in order to make today possible, this competition could only be done with the help of judges. So thank you so much for our judging panel who will be judging our 8 students at this 3-minute competition. We have, if you'd like to please raise your hand, they're going to be sitting in the back row when I call your name. We have Daniel Ivanko. He is the Director of Journal Operations and Systems at AACR and a Jefferson alum. So thank you for joining us today. We also have Timothy Huff, who's the Product Development and Director at Hello Insight. Thank you for joining us today. We have Crystal Kraft, who is the research lead for advanced therapeutics at West Pharmaceutical Services, also a Jefferson alumni. Thank you for joining us. And we have Carolina Pombodima, who is very close to being here. I promise she's going to be here. She's an accounts manager at Alpha Biocom. And we have Liz Tung, who is a health and science reporter at WHYY's The Pulse. If you heard her the radio show, she is part of making that happen. So thank you for joining us today, Liz. Hi, Liz. So those are our judges. The rules of the competition are as follows. So it's an international competition. As such, there are very strict rules that we have to adhere to because, as I'll state later, the finalist is going to move on to the regional competition, and then that finalist will move on to the national competition. So our students will be competing, and the rule is they have to do their talk in 3 minutes, spoken word, no singing or anything. And a presentation begins when they start speaking or moving, but they can get to their spot. They don't have to start from their seat. In addition to that, they have a 1-slide requirement, no animation, and no props. And the third major rule is that this is for a non-specialist audience. And that is why we encourage everyone to bring their families and friends, because I know personally a lot of people are always like, what do you do? And I don't know what to tell them. And so I personally wanted my mom and grandma to be able to understand what I was talking about. And I think a lot of the students here today felt the same. And then the judge's decision is final. So what does that mean? What are the criterion for the judges? So I'm not going to, go through that. But the students will be judged on comprehension and content. Equally, they'll be judged on engagement and communication. So for the comprehension and content part, I'll just briefly go through it. What the judges are looking for is that the purpose is clear, that the student speaker is stating that significance and impact as clearly as possible, has as little jargon as possible in their talk, and it's very accessible to this audience. What we're not looking for is a talk for other scientists, a poster presentation, a research paper, and again, we are not judging the science, we're judging how well you're able to communicate the science to this audience. For the engagement and communication aspect, the judges are looking for that the student is enthusiastic, that they have a lot of stage presence, that the PowerPoint, it's accessible and supports the presentation. You'll see they all have a variety of different PowerPoints to support their work, and it's very interesting how they chose those to go along with their talks. And also that they had a clear hook that makes you want to know more. That's very important. What we don't want to see is, what we're not judging them on is their choice of metaphor. We're not judging them on the blurb that's in that some of you have been able to see, the 100-word summary of their talk, or you can get to it by a bit.ly that's in your program. You just follow the link on your phone. They're not being judged on that, though. They're being judged on the presentation. And also, they're not being judged on their research topic. If their research is failing, they're not going to not pass this award. It's, the point is that they can explain well what they're doing and why it's important. So what will they win? So thanks again to the Graduate College of Life Sciences for supporting these prizes, because nothing makes anyone want to do anything more than getting paid for it. So the first place winner will win $1,000 and will be entered into the NAGS Regional 3ONT competition, which is the National Association for Graduate Students. The second place winner will win $500, the third place winner will win $250, and the People's Choice will win $250. And anyone that wins one of those three could also win the People's Choice. It's free for all. Oh, okay, so I'd like to say lastly, this also could not have been made possible without the support of the Center for Teaching and Learning. They have been instrumental in supporting us in preparation. Thank you to Pam Walter, yay, who is... She worked tirelessly with many of our students to get them where they are today in one-on-one training sessions using resources that are available in the Center for Teaching and Learning, as well as Liz D'Angelo, who is somewhere in here. Thank you for our marketing help. That was very, very, very necessary. And look at this room, so full. So without further ado, we're going to get started. Does anybody have any questions before we start? Yes. Yes, the People's Choice will occur after the entire competition ends. While the 5 judges go to deliberate, you will be texting your People's Choice into your phone, into the computer that Rogan has. Poll everywhere if anybody's used it before. It's a way to get data from lots of people in a classroom. So if you have a phone, it doesn't have to be a smartphone, it can be a dumb phone, it's okay. You can text it and it will be judged that way, but I'll explain more of that after the competition. Any other questions? Okay, so without further ado, I'd like to bring up the first speaker, and that is Alison Shupp. Please come up. Okay, so Alison Shupp is in the Genetics, Genomics, and Cancer Biology program in the lab of Dr. Karen Bussard, and the title of her talk is Cancer Metastasis, What's Bone Got to Do with It?
Did you know many breast cancer patients may be unaware they have metastatic cancer, which is when cancer spreads to other organs? Traditionally, patients with breast cancer are given chemotherapy to kill their tumor, and then they go about their lives living disease-free, or so they think. It probably will not come as a surprise that cancer cells are sneaky and they can avoid being killed by chemotherapy. Instead, these cancer cells can spread to other organs in the body, and instead of rapidly growing and forming a tumor, these cancer cells can lay dormant. Cancer cells can survive in a dormant state for years or even decades, but cancer cells are not usually dormant forever. They're just waiting for the right time to grow and can cause a patient to relapse unexpectedly. One common site for cancer cells to spread and lay dormant is the bone. One way to think about cancer dormancy is to picture a seed planted in soil. So imagine you're planting seeds in a garden. You'll water them and wait for a plant to grow. But if summertime comes and a plant never grows, it could be because you had bad seeds, or it could be that the conditions just weren't right. Maybe the soil wasn't fertile. In this analogy, think of the cancer cell as the seed and the different organs of the body as potential soil. When cancer spreads throughout the body, there is a potential for the cell to rapidly multiply and form a tumor. Or the cancer cell may lay dormant, waiting until the conditions are just right or the soil is fertile to grow. This theory about seeds and soil may explain why cancer cells spread to the bone, but in some instances do not rapidly grow. Maybe the bone is not a fertile soil. In my research, I'm investigating what is unique about the bone that in some patients results in cancer dormancy. I found that a specific cell in the bone produces factors that arrest cancer growth. These factors are called extracellular vesicles. Vesicles are small membrane-bound structures, like a small bubble produced by cells. Vesicles can be produced by one cell and taken up by another, and in this way, they act like small packages to deliver molecules like DNA, RNA, and protein between cells. I found that cells in the bone produce vesicles that are taken up by breast cancer cells, and when this happens, it arrests cancer growth. Many researchers are now investigating ways to harness the power of vesicles to treat cancer, and I am investigating what is unique about the vesicles derived from the bone that is able to arrest cancer growth. I hope to identify molecules that can be utilized in cancer therapy, and hopefully we can keep these cancer seeds from growing. Thank you.
Thank you, Alison. Next up is Emanuela Dylgjeri, and she is in the Biochemistry and Molecular Pharmacology program. She's in the lab of Dr. Karen Knudsen, and her title of her talk is The Strange Case of Dr. DNAPK and Mr. DNAPK.
You may have heard of the strange case of Dr. Jekyll and Mr. Hyde. But in case you haven't,
Dr. Jekyll was a well-respected researcher, and Mr. Hyde was an evil stranger that appeared out of nowhere and started causing trouble. Well, in the end, it turns out that Dr. Jekyll and Mr. Hyde were one and the same person, switching back and forth between two extreme personalities, one good and one evil. In my research, I'm dealing with a similar character called DNAPK. In healthy people, DNAPK is an essential do-good protein that repairs cells when they're damaged, it gives them instructions to stay fit, and it provides them with just the right amount of energy for them to run like a well-oiled machine. Dr. DNAPK, it's perfect. However, in cancer, DNAPK morphs into the diabolical Mr. Hyde, and that's where my research comes in. You see, I study prostate cancer, and most people think that it's an old man's disease and it's easily cured. Not quite. Did you know that every 18 minutes, a man in the United States dies of prostate cancer? That is a son. an uncle, a grandfather, a father, every 18 minutes. What am I doing to help? For that reason, in my PhD, I have two goals. One, figure out how evil DNAPK works in cancer, and two, find a potent drug to suppress its functions and make the patients live longer. For how it works, I have figured out that evil DNAPK in cancer gives the cancer cells a carbo load, providing them excessive amounts of energy so they can grow faster and spread to other organs. I've also found out that DNAPK has partners in crime, and they're just as evil. They help cancer cells be so strong to resist chemotherapies and become the merciless killers of our loved ones. Using that knowledge, just like Dr. Jekyll set out to find a potion to kill Mr. Hyde, I set out to find a drug to kill DNAPK in cancer. And I found two drugs that when they are combined together, one new and one the patients already take, they kill cancer cells faster in the laboratory. And now, it's being tested in prostate cancer patients. I believe that understanding how DNAPK works in cancer is the way to defeat it. And it is my mission and my hope that through my research, I'll be able to help those unfortunate patients with too much DNAPK and provide them a personalized treatment that will help them live longer. And maybe, just maybe, we'll defeat DNAPK and make sure that it never turns into Mr. Hyde ever again. Thank you.
Thank you, Emanuela. Next up is Christine Fisher. She is in the Immunology and Microbial Pathogenesis program. She works with Dr. Matthias Schnell, and the title of her talk is Ensuring Against Rabies with a Broader Vaccine.
Vaccines are a lot like insurance. Both are proactive steps that we take to prepare ourselves for the unlikely but possible event of something bad happening. And insurance companies like to remind us that our policies may not always cover every predicament that we could get into, such as raccoons getting into your roof or a car accident caused by your blind spot. And like insurance policies, sometimes vaccines don't protect against everything that you would expect them to. One vaccine where you really don't want to miss any blind spots is the rabies vaccine. Rabies is a disease that's spread by wild animals, and it kills 60,000 people around the world every year. That's about the same number of deaths caused by the flu in the US every year. But to really put those numbers into perspective, millions of people get the flu every year, and they recover and survive okay. Only 14 cases of survival from rabies disease have ever been documented. Fortunately, there's a vaccine, as I mentioned, but here's the blind spot. There are actually 17 different viruses that can cause rabies disease, and the vaccine was only based on one of them. Some of these viruses are so different that the vaccine does not protect against them, but they're still deadly. So my thesis work is focused on making a vaccine that can protect more broadly against more of these rabies-related viruses. So I'll start with the current vaccine. The most important part for our body to recognize is the outer layer of the vaccine. It's the part that looks like the little beads on the picture. And what I did is I re-engineered these little beads to contain parts of two different viruses. So 2 is better than 1, but I did this strategically, and my goal is actually that it will protect against most of these viruses. So you can think of it as a vaccine that is greater than the sum of its parts. When I put this vaccine into mice, they developed strong immune responses in response to it. It was a very good sign that the vaccine was working. But even more importantly, they were completely protected from disease from 2 very different rabies-related viruses, just as we had hoped. So it's a very important first step, and the next steps will be to test even more of these viruses to see just how broadly protective this vaccine is. These rabies-related viruses are pretty rare, but they're deadly. And if there was an outbreak and we had no vaccine, it would be devastating, similar to other outbreaks of rare infectious diseases that do come up from time to time. Through my thesis work, I hope to ensure that just in case we have a vaccine ready and that we're in good hands. Thank you.
Thank you, Christine. Next up is Nicolette Heinsinger. She's in the Neuroscience program. She works in the lab of Dr. Angelo Lepore, and the title of her talk is Relieving Chronic Pain, Silencing the False Alarm.
So who here has ever stubbed their toe or fallen and scraped their knee or accidentally touched a hot stove? I can bet that none of those were fun experiences. Now imagine if you randomly felt burning or pins and needles sensation or felt pain when your clothes brushed against your skin. That is the reality every day for up to 80% of spinal cord injury patients. Unfortunately, most medications either don't work for all patients or, like opioids, have severe negative side effects. Needless to say, we need better treatments for patients that are both safe and effective. So why is chronic pain caused by spinal cord injury so resistant to treatment? You can think about the flow of information from your body to your brain like a relay. You touch a hot stove, heat sensors in your fingertips will send that information to your spinal cord. And your spinal cord will collect that information and relay those signals to your brain. This system works like an alarm clock, because the second that your spinal cord receives those messages that your hand is in danger, it's immediately going to pull it back to prevent further damage. And your brain is going to remember that unpleasant, painful feeling and make you not want to touch that hot surface again. Just like an alarm clock prevents you from oversleeping and missing work in the morning, this system works to prevent damage to your body. However, after spinal cord injury, this system gets a little confused, and the alarm clock will start going off at times when it's not supposed to. Now, information such as the light touch of a sweater against your skin will be sent to the spinal cord, but this time those pain signals will become amplified, and your brain will start to receive more pain messages than it normally should. What I'm trying to do is figure out why this alarm clock keeps going off even in the absence of a painful stimulus, and then figure out a way to silence it and reset it so that it only goes off at the proper times. In our lab, we have identified elevated levels of a protein called FB2 in the spinal cord after injury. And we believe that this protein is partially responsible for those amplified pain signals to the brain. In my research, I'm using a new therapeutic approach called gene therapy to decrease levels of this protein in the spinal cord. And then I can use various behavioral assessments in order to measure that decrease in pain after this treatment. Ultimately, the goal of my research is to silence those false alarms and provide spinal cord injury patients relief from their chronic pain. Thank you.
Thank you, Nikki. Next up, we have Diego Marin. He's in the Genetics, Genomics, and Cancer Biology program. He works in the lab of Richard Scott. And the title of his talk is A Test That Puts Art on the Fridge.
So first of all, I would like you to take a look at those two fridges. I don't know you, but for me the difference is obvious. When I look at that one to the right, the colorful one, I can even listen to the screams and yelling of go to your room or do your homework. Now the other fridge, on the other hand, the empty one, silence and peace. But that empty fridge could also tell a story, and a very tough one. For example, a story of a negative pregnancy test after the other one, or probably constant failure of in vitro fertilization cycles. And it could be tougher, it could be more painful. For example, recurrent miscarriages or pregnancy losses. And it is very hard to understand how hard it could get, especially for people who already have children, or someone like me who's just busy building our career right now. So, but for those of you who have that colorful fridge at home, or whoever had it, could you picture it empty? Well, that is the case of 15% of our population, meaning that 1 in 7 of us will face that problem eventually. Now, infertility affects both men and women equally, and it gets worse the older we get. But for women, it's particularly difficult because if they want to spend more time building a career, or maybe postponing motherhood after 6 years of grad school, it's not easy. So what can we do to fix that problem? At least I focus on that guy in the middle, and that is how you and I look like when we were 5 days old. We call it an embryo. So as beautiful and well-shaped as it may look, because by experience I can tell you, this one is a very good-looking one, about 80% of these embryos are abnormal and defective by the time a woman reaches 42 years of age. So my whole work and the focus of my work is to try to understand what's wrong with that embryo. Why did it result in a miscarriage? Or why didn't it end up in a healthy pregnancy? And by literally taking and analyzing small pieces of it, I've been working on developing a test that assesses not only how healthy the embryo is, but also its fitness, so that we can gather that information and we can select the one best embryo to be used in an infertility treatment. So by just simply doing that, not only have we dramatically decreased the risk for miscarriages, but we have also shortened the time of treatment, which is not a pleasant one. But most importantly, we have significantly increased the chances from getting from this fridge to the other one. Because the more we understand those guys in the middle, the more art we can put on a fridge. Thank you.
Thank you, Diego. Next up, we have Nicole Wilski. She's in the Immunology and Microbial Pathogenesis program. She works in the lab of Dr. Christopher Snyder, and the title of her talk will be Friend or Foe, Using Viruses to Cure Cancer.
Hi everyone, can I get a show of hands? How many of you have seen Jurassic Park? Great, the original one, right? Not the one with Chris Pratt? Okay. So for those of you who haven't, or for those of you who need a refresher, there's one main scene that sets up the science for the entire movie. And in this scene, everyone sits down in the theater and watches a video where this guy, this anthropomorphized piece of DNA they call Mr. DNA, explains, fossilized tree sap, which we call amber, waited for millions of years with a mosquito inside until Jurassic Park scientists came along. Using sophisticated techniques, we extracted the preserved blood from the mosquito and bingo, dino DNA. Now imagine for a second that you're all Jurassic Park scientists. You extract the DNA that you expect to be from a virus, or from a dinosaur, sorry, but it's not. It's from a virus. It'd be a much less interesting movie, right? But the truth is, viruses are prehistoric pests that have been around since at least the Jurassic period. And ever since humans came along and realized that there are things out there trying to make us sick, we've been trying to stop them from doing so. From the flu that can knock us out for a few weeks in the winter to even smallpox or polio, stopping the spread of disease is undoubtedly a mammoth scientific achievement. But what if viruses weren't always the enemy? For my thesis, I've been working with one of these ancient viruses called cytomegalovirus, or CMV. CMV actually infects 90% of us by the time we're 80 years old, and it sticks with us for life. But because it doesn't normally cause any symptoms, we'd never even know we had it. The incredible thing is that we can use CMV to our advantage when it comes to cancer. If you take a virus that's weakened, it can't spread, it can't cause disease, and you put it into a tumor, it stops the growth of the tumor just like that. And if you continue to put the virus in over and over and over again, it will continue to stop the growth of the tumor as long as there's virus around. And during the course of my thesis, I've been able to track down the specific cell in your immune system that's responsible for this effect. This particular immune cell is actually infected by this virus. And once it's infected and senses that it's infected, it can signal to the other immune cells to come to the site of infection, and it essentially stops the growth of the tumor in the process. So what we're saying is, instead of the virus and the immune system being at odds, the virus can actually work with your immune system in order to stop cancer from growing. It's an incredibly promising technique that can be used in combination with therapies that are currently on the market, especially in patients that aren't responding to current therapeutic options. So promising, in fact, that I think it's time we all ask ourselves, viruses, are they always the enemy? Or could they be our friends? Thank you.
Thank you, Nicole. Next up, we have Brandie Morris. She is in the Neuroscience program and works in the lab of Dr. Piera Pasinelli. The title of her talk today is Tidying Up Toxic Proteins in ALS.
Imagine you're waiting at a crosswalk. You begin crossing the street when you stumble. You would probably attribute it to clumsiness and ignore it, but then it happens again and again. With time, you become weak and lose the ability to move your limbs. As your muscles begin to waste, you have trouble swallowing, can't eat without help. Eventually, you can't breathe without a machine. All the while, you're aware of what's happening to your body, but you can't do anything to stop it. Now imagine that the time between that first stumble and death can be as little as 2 years. This is what happens in ALS. ALS, or amyotrophic lateral sclerosis, is commonly called Lou Gehrig's disease. You may know it as the ice bucket challenge disease. What you may not know is that 15 Americans are diagnosed with ALS every day. One of these people was my aunt who passed away in 2010. Her first stumble happened walking the streets of New York, and she, like anyone else, just thought it was clumsiness. ALS develops when the brain cells known as motor neurons communicating between your brain and your muscles die. I study one of the more common mutations that causes ALS. In this particular mutation, the protein-making factories of the cells go rogue and produce these unexpected proteins. Now, it's unclear if these proteins are the cause or simply passengers in the disease course. The formation of these proteins is similar to putting together your own furniture, let's say an IKEA chair. Your instruction manual informs the formation of the chair, much like RNA informs the creation of protein inside a cell. Now, you're eager and you want to work quickly, so you don't follow the instructions very closely, instead just skipping to the middle of the manual or leaving a few steps off at the end. What you get is something that looks like a chair, but its haphazard construction creates an unintended product. And in the case of the brain and spinal cord, these proteins or your furniture can have toxic consequences. They can take up space in the cell they shouldn't and interfere with the cell's normal functions, creating an environment that probably doesn't spark joy. Now, a lot of people like to blame the instruction manual, but what if ALS could be treated simply by cleaning house? The goal of my thesis is to understand how these unexpected proteins affect the overall health of motor neurons and muscle. I create models where I can look at these proteins without other confounding factors and see what changes occur as they would in ALS, such as cell death or movement deficiencies. Our lab has been able to demonstrate that these proteins can be toxic to isolated cells. So the next step is to see what happens to networks of cells as well as animals. Not only will these models help in our understanding of what these proteins do, but they'll also help in screening potential therapies to treat this ALS. And if treating this ALS is as simple as tidying up and eliminating the toxic proteins, maybe we can help change a few of the 15 diagnosed ALS patients each day into ALS survivors. Thank you.
Thank you, Brandie. Thank you very much. Next up is Lindsey Mayes. She is in the Biochemistry and Molecular Pharmacology program. She works in the lab of Dr. Emad Alnemri, and the title of her talk today is Flipping the Switch. How can we stop inflammation before it goes too far?
I'd like you to think about the last time you had a cold or the flu. You probably experienced some coughing, sneezing, maybe even a fever. Well, all of these symptoms are actually methods your body uses to fight an infection. For example, a fever heats up your body temperature ever so slightly that it kills the virus or bacteria living inside the body. Now, these symptoms are due to inflammation, which is basically an emergency flare alerting the body that it's been infected and it needs to defend itself from this infection. Luckily for us, though, after a couple of days, the fever goes away, the coughing stops, the inflammation has effectively removed the infection, and you can go on with your normal life because the inflammation has turned itself off. But what if this inflammation doesn't turn off? Although inflammation is important in fighting infections, as I've just described, excess inflammation contributes to a wide variety of diseases such as Alzheimer's, arthritis, diabetes, and cancer. So it's extremely important that we have a better understanding of how inflammation works. But unfortunately, this is still very much a mystery to scientists today. That's why for my PhD research, I'm trying to better understand how inflammation is regulated. How can we flip the switch and turn off inflammation before it goes too far and progresses to a chronic condition? We can study this in the lab in a petri dish with immune cells and infect these cells with different viruses or bacteria and measure the infection and immune response. Through these experiments, I've actually discovered a protein that's important in regulating inflammation. Think of a grocery store right before a snowstorm. You probably have a large influx of customers coming in, rushing to get their last-minute bread, milk, and eggs. And in response, the grocery store manager can open up additional checkout lanes, so that way the extra customers can flow through freely and get home safely. When there's less customers, they can close those extra lanes. The protein that I've discovered is kind of like the grocery store manager, because it can sense when there's an imbalance between the inside and the outside of the cell. In response to this imbalance, the cell is able to open channels, or grocery store checkout lanes, to allow for better flow through from the inside to the outside of the cell. When the cell is balanced, it's happy, and happy cells are actually able to turn off this excess inflammation. This is really exciting because we can use this protein as a target for therapies to flip the switch and prevent inflammation before it progresses to a chronic condition. Thank you.
Thank you, Lindsey. Thank you so much. Can we please have another round of applause for all of our contestants today? So the judges are going to go out into an area out the door, and they're going to deliberate and score their sheets after all these amazing presentations. I know it was probably a lot to take in for everyone in here, so we want to give them some time to reflect and make the best choices in scoring our students here. While that's happening, we're going to have the People's Choice Award. That's all of you. Oh, I just shut that off. The People's Choice Award. So the People's Choice Award is for people like you who are in this audience and are not the judges. You're going to also judge the students' presentations on comprehension and content and engagement and communication. So what you're going to do, and don't worry, this will be on the next slide, but you're going to text this very long code to the number 22333, and after it prompts you, you'll choose your choice. So what's that look like? So on this slide, it'll hopefully help guide you. You also have your your programs might help you too. And when you do that, you just type what I just said, and you choose, each one of these people has the following letter associated with them. So feel free to do that when you're done. Walk around, enjoy, talk to whoever you like. We're gonna take a break for about 10 to 15 minutes. Thank you. Okay, the GSA president is in the house. Thank you for coming to bear us with the certificates of the People's Choice will be announced first, then the third place winner, the second place winner, followed by the first place winner, is the winner. But everyone is a winner, and we really are proud of all of you guys here. You all put in so much work, and I think you've all said to me how impressed you are of each other and how everyone was really, really blew you away. And I hope everyone here today learned some things and had a good experience. So Rogan is going to be standing in the center. When you get the award, please come in front and do the whole shake hand thingy when you get your award, and Dr. Kozlowski will take a picture of you guys with Rogan. Okay. Get in the center. Oh, I need to know who it is. Okay, yes. Okay, so for the People's Choice Award, drum roll. Ooh, I like this. Okay. The winner is, this is your choices, everybody, Nicole Wilski. Yep. And then smile for Dr. Lisa. Wait. Thanks. Thank you. Yay! Oh, so loud. Thank you, Nicole, for teaching us about viruses and cancer and Jurassic Park. Okay, for the third place winner, drum roll. Wait, I didn't see it. The winner is Christine Fisher. you for Christine for sharing about insurance and rabies with us. All right, the second place winner, drum roll, is, what was it again? Okay, Brandie Morris. Thank you, Brandie, for teaching us about putting IKEA furniture together properly and how important that is for our brains. All right, and for the first place winner, this is the final winner, and this winner will move on to the National Association of Graduate Students Regional Competition. Drum roll. Nicole Wilski. Thank you everyone for participating. Thank you. Good job. And thank you again for teaching us things. Everyone here, thank you for all of your participation. Thank you for attending. Thank you for your support. We have a reception out those doors right there by the stairwell. Please come up and talk to all of the contestants, and thank you for joining us in the Science Communication and Outreach Initiative. Take care.